Welcome back. This is our last video on microfilaments, and what I'd like to talk about here are how cells respond to growth factor signaling. And we're going to obviously look at the response of the cell in terms of its microfilament network. So the cytoskeleton of cells can undergo dramatic changes in response to signaling growth factors, and many of those signals that come in from growth factors act through a family of monomeric G proteins called the Rho GTPases. And so I hope you remember from cell signaling in general biology that G proteins are proteins that bind GTP, and many of them are involved in cellular signaling. So the three key family members of the Rho GTPases that regulate the actin filament network are Rho itself, RAC, and a protein called CDC42. So if we look at these cells here, so these are confocal microscopy images, fluorescence microscopy images, where we're looking at the actin filaments in cells. So here is a cell which is serum starved, and when a cell is serum starved and just growing in a dish, and there's no serum in the growth media, that serum is usually what contains all of the growth factors that a cell is going to respond to. And many cells can't exist in a serum starved state for too long, but you can keep them in a serum starved state for short periods of time. So we can see, not much organization of the actin network, maybe mainly in the cortical actin just under the cell membrane. Now, if we activate Rho, we see that stress fibers form, those kind of thick bundles of actin filaments that form stress fibers. If we activate RAC, then we see the formation of lamellipodium. So that would be the stimulation of a migratory behavior and structures required for that. And if we activate CDC42, then we see the formation of lots of filopodia. Those are the very thin finger-like projections you often find at the end of lamellipodia. So let's just look at the Rho GTPase and look to see how that works. So Rho GTPases are stimulated by a group of proteins called guanine nucleotide exchange factors, or the GEFs here. So what the GEF does is it takes Rho with GDP bound and it kicks out the GDP and allows Rho to bind GTP, and that activates the Rho protein, and then Rho initiates a signal transduction cascade that stimulates actin remodeling, so that we get, in the case of Rho, we get the stress fiber formations. If this GTPase here was not Rho but was RAC, then we would get remodeling to form lamellipodia, and if the G protein were CDC42, then the signaling would lead to actin remodeling to produce filopodia. So the GEF here switches on Rho. Now Rho is sequestered at the membrane, and so it's a lipid anchored protein and it's bound at the membrane. So Rho will turn itself off with the assistance of a protein called a GAP. So if you think back to what you know about G proteins, after a short amount of time, they will spontaneously hydrolyze their bound GTP to GDP, and that will make them inactive. And you can stimulate that by the action of a GAP protein. So that's a GTPase activating protein. So the GEF and the GAP work against one another. So the abundance of these two and the location of the cell membrane will either give us an abundance of active Rho or an abundance of inactivated Rho. We can also then sequester the Rho in the cytoplasm, so we can remove it from the membrane and put it in the cytoplasm. Now, to be involved in signaling to lead to actin remodeling, we must have Rho at the membrane. So if we pull it away from the membrane using these proteins called guanine nucleotide dissociation inhibitors, they then sequester the Rho in the GDP bound form in the cytoplasm, and that means you cannot stimulate the actin remodeling via the Rho protein or RAC or CDC42. So there's a little bit about how cells respond to growth factors using these small soluble G proteins. And when we come back, we will be looking at intermediate filaments, and that will be pretty short. I'll see you soon.